morning, everyone. Welcome to this Food Techie Sherpa series, which is organized by AFST Mumbai chapter in collaboration with CAS MB and ICT Mumbai. So this is the 12th talk in the series, and I'm so happy to see the enthusiasm shown by all our students. And I'm very sure that all of us are been enlightened from listening to all these stalwarts from the industry, academia, and the regulator. So before we start with the lecture, I would like to just give few minutes to the introduction of the AFSTI. So the AFSTI, that is Association of Food Scientists and Technologists India, it is one of the oldest association which is established in 1957 at CSIR CFTRI Mysuru, and here at Mumbai chapter, we can say it is one of the biggest chapter of the association and the most vibrant and dynamic chapter. And this lecture series, that is Food Techie Sherpa Edu series, it has been scheduled in a row, that is second and the fourth Saturday of every month, and I every month. And in general, if we see about the AFSTI and AFSTI Mumbai chapter in particular, the major goal or the idea of the AFST is to create a common platform where the stakeholders of the foods come together, can deliberate and take the things forward. So here different, like the regulators, academicians, students, industry, all of them come together and talk to each other and take the things forward related to the foods. And when we talk about the AFST, and because this is the platform, we can say that majorly for the students, what are the benefits to the student? One of the major benefit is the networking. The students can network with the industry, the regulators, and get the things forward. We have the poster and the paper presentation where the students can come forward and do the presentation. Currently, we will be having the ICFOST 2024 at DY Patil, and I'm sure many of you must have already registered, are or going to register for the conference. And we can say that it is going to the Kumbh Mela of the food related students and the industry and the regulators. So along with that, we do have the mentorship programs, we have the academic guidance program, different award functions. So many of you must be there for the 2 days before the World Food Day, where we had Dr. K.U. Naram Award. So where the first and the second rank holders of the different institutions from the Mumbai are being awarded. Then likewise, we have the different initiatives which are related to the, as I said, the ICFOST is going to be there in the December 2024. We have different regulatory summits. We partner with the different events like FIHI, Annapurna, which is Anuka Select now, then US Chickpea Council, US SAG, Dairy, and others. And the flagship programs, if we see about the AFSTI, as we everyone knows that the Nutrition Week, or nowadays it has been called as the Nutrition Month celebration, World Food Day, World Food Safety Day. So with this, I thank each and every one of you to listening to the intro of the AFST, in particular to the Mumbai chapter. And with this, we move forward for the lecture, where today we have Dr. Geeta Ibrahim with us. And just before yes. So, Dr. Geeta ma'am, we are so happy and honored to have you with us for this lecture series on the topic application of bioactive peptides in food industry. Dr. Geeta ma'am is she has retired as a principal of College of Home Science, Nirmala Niketan. She has teaching experience of more than 42 years. She has been recognized guide for the PG teacher for the MSc Foods, Nutrition and Dietetics. She is recognized guide for the biochemistry at University of Mumbai, and for the nutrition also at the University of Mumbai. And though we are saying that she is a retired as a principal of the Nirmala Niketan, post-retirement also she has been very active. She is the core committee member of the Spina Bifida She is a CDC member of Walia College of Commerce, IQAC member of St. Xavier's College. So I would say retired is the only word, but she is more active after the retirement also. So with this brief introduction, I hand over the stage to Geeta ma'am. Geeta ma'am, once again, thank you so much for accepting our invitation and delivering the talk for this Food Techie Sherpa series. Thank you
Thank
so
you.
much.
Thank you. Thank you, Tripti, for this wonderful introduction. Actually, I'm not, very little of it is with me now. I'm really a retired person. So it's a great pleasure and good morning to all of you across the country. And thank you so much. I'm so delighted to be here. At the outset, I wish to thank AFSTI Mumbai chapter for giving me this opportunity to be here on this platform and to be sharing the Food Techie Sherpa platform, I should say, and
Yeah.
for sharing a small presentation on the bioactive peptides. I thank Ms. Chinmay Deul Gaukar, Mr. Nilesh Lele, Ms. Samiksha Pawar, Ms. Jyoti Gokhale, Ms. Sukhada for this wonderful and innovative method of reaching out to students across the country on relevant and updated topics of food science and technology. You don't know how lucky you students are that sitting at home or wherever you're sitting, thanks to the COVID period, one of the positive outcome of COVID is this online teaching. And through this method, we have been able to, get the best and from across the world, I would say, global platform to come together and share very important topics, which otherwise would not have been possible. So I congratulate the AFSTI and ICT for this innovative approach. It's been a long association with AFSTI Mumbai chapter and having met most of their members over a span of almost 30 years or so. Every interaction has been a learning experience for me.
Yeah. Yeah. Now it
Yeah.
is okay.
So I just want to again thank, as I was saying, my academic interaction with AFSTI Mumbai chapter and its members is a very old association of more than 30, 40 years. And every interaction has been a learning experience for me. I also thank them for their support in all spheres of student activities, whether it is scholarships or organizing lectures or mentorship programs. They're always present and they have been coming to different institutes, to our college in particular, Nirmala Niketan, when I was there, first as a teacher and then as a principal. So it's always a reach out to them and they are there and organizing all different kinds of programs for the benefit of students. It is a very good interaction for me and academicians to be interacting with the industry, which is the need of the hour. And that is what AFST provides. Even yesterday, I have attended a very good session by PFNDI and where I met so many of the FSTI members. And it was such great pleasure to be, back on this platform. And I hope I can remain more connected with you all. It is wonderful. I don't know who are the students from which parts of India or part of Bombay. I don't know, but I'm always very happy to connect with all youngsters. It makes me feel young and keeps me young. I can see Jyoti smiling. So yes,
Yes,
I feel
true.
young being here on this
Yeah.
wonderful platform. And let me go ahead. I think if we can share the PPT because I don't want to take too much of your time. And I hope I can deliver whatever I have with you to you in a short span with a little knowledge that I have gathered on this topic, which is a little new to me, of course, but interesting. That's what I said. It's a learning experience for me. Every interaction. Traditional biochemistry is one thing, but when it's application of biochemistry, I'm thankful to organizations like them who give us this opportunity to study again. So learning is a lifelong experience. So over to you, and if I can have my presentation,
Yes, ma'am.
I'll be happy.
Sure, sure,
Thank you
ma'am.
so much. Thank you.
I request Astha to share the ma'am's presentation, and
Yes.
ma'am, she will be there with you for the presentation related
Okay.
things.
All right. So I have about 45 minutes for the presentation.
45 minutes to 1 hour is fine, ma'am.
All right. Okay, fine. Thank you. Thank you so much.
So before we start with the presentation, I request all the participants to be on muted. If you have any questions, you can write in the chat box and any one of us from would take the questions at the end of the session. Thank you so much.
Thank you. Thank you. Okay, so let us begin with this very novel topic. I know you are all postgraduate students, I've been told, and so some of the basics of food science and chemistry and biochemistry is known to you all. So there would be certain terminologies which I may not go into details with, presuming that you are already aware of the meaning of these terms. So the topic as such is the application of bioactive biopeptides in the food industry. So let's go ahead. Now, why this interest of this particular topic? Because as it is, when you look at the philosophy of science, what is science? It is all to do with improving the quality of human life. And when we talk of food technology or food per se, we look at food not only from the eating experience or giving us the dietary experience, but nowadays the food is also examined for their bioactive compounds, which, when consumed by themselves individually, isolated, can be beneficial to human health. So the rising demand for these sort of foods has led to the search for alternative, sustainable, and multifunctional sources. One of them being peptides. Now, peptides, as you know, come from proteins. So, where do you get these peptides from? From the agro-industrial waste, which are and the byproducts from very unknown, very strange sources like insects, marine and plant enzymes, which could be the future sources of these bioactive peptides. So they are derived from different proteins. They are biologically active regulators that can be used as food additives. And why are they used as food additives? It is mainly to prevent oxidation and microbial spoilage of foods. Proteins, are large molecules with a high molecular weight. Their absorption and digestibility is a little difficult in the human body. Compared to them, the peptides are simple in structure. They are more stable with a low molecular weight. And therefore, food proteins are chosen to isolate these bioactive peptides. Why do we think of isolating them? One of the reasons is to add value to the waste that is otherwise going to be like, the industrial waste. It's a value addition to that. So you extract the biopeptides from there. And then with these biopeptides, the sequences of the amino acids which are present in these peptides, because now, as you know, proteins are nothing but a sequence of amino acids. So when you hydrolyze them, the smaller peptides are also going to be composed of amino acids, but in smaller amounts, maybe even ranging from 2 to 20 or 30. And these amino acid sequences have specific benefits in all different spheres like cosmetics, food additives, nutraceuticals, pharmaceuticals, antimicrobials, antithrombotic, etc., etc., mainly because of their specific sequence, their action, and the structural diversity. So what are these bioactive peptides? As I told you, they are amino acids that are typically maybe even two amino acids together may have some activity, or maybe there could be a stretch of 20 to 30 amino acids, which are definitely joined by the peptide bonds. That is the backbone structure of any protein. And they are encrypted within the plant protein structure. So you need not plant, but the parent protein structure, for which we need to extract them out, and where we will go ahead. When we go ahead, you will learn the different extraction procedures. So most of them, the biopeptides, have shown the presence of amino acids like proline, arginine, lysine, and many other hydrophobic residues. Next one, please. In the native sequences, they do not show much of biological activity, but they can be activated by enzymatic, chemical, or microbial hydrolysis. Of them, enzymatic hydrolysis so far is supposed to be the most effective one. And the peptides which are formed from these enzymatic hydrolysis, they can be easily absorbed in the intestinal tract, as well as they can enter into the bloodstream. Now, any sort of procedure, the ultimate aim would be to see how well it is absorbed, how well it is tolerated, and how well it is effective in the human body. So enzymatic hydrolysis, enzymes themselves are proteins, and if they can bring about hydrolysis without much causing any damage, they are one of the best ways of breaking down the proteins. So these, what happens when you break down the proteins to smaller peptides is that they decrease the antigenicity. Now, proteins by themselves may bring about some antigenic reactions in the body. But when you break down the protein to smaller peptides, their antigenicity decreases. So that antigen-antibody reaction may not be that adverse or may not be that severe or may not be there at all. And therefore, they can be used even, they are being used in infant formulas, the peptides. It's not just that you have about 20 to 30. There are some longer peptides also, which have been reported to have about 43 amino acids, mainly from soy proteins, which have shown to have anti-cancer and cholesterol lowering properties. One of them is lunacin. Okay, next one. What makes them active? It is said to be the hydrophobicity. As we have seen earlier, it's mostly the hydrophobic amino acids which are playing a key role, and these are the ones that are interacting with other biological molecules. So the bioactive peptides, they have specialized target activities on the target tissues and nowhere else. So as a result, what happens? A concentrated action on specific target tissues decreases the toxic effects which would have otherwise happened. You take some drugs, and we know that synthetic drugs do have some side effects, though they are very effective, and they are known to be excreted also. There is what is called as ADME of drug metabolism, which you may have studied. But there could be a possibility of side effects, whereas the peptides, they will go and just act on the target tissue, and otherwise there will be no other side effects or even causing any environmental damage due to their excretion. They prevent, by and large, they prevent the lipid oxidation by scavenging free radicals. They chelate the metal ions. They prevent enzymatic degradation. They prevent microbial growth. And the ultimate aim, of course, in the food industry, if you're looking at besides their action in the human body, in the food industry, it is mainly to maintain the shelf life of developed products. Why food industry and why the bioactive peptides are gaining so much importance? Because food safety and spoilage prevention are the main concerns in the food packaging industry. That is one of the greatest concerns. And therefore, food packaging is now becoming more and more innovative. So it is a protective. What is food packaging? It is a protective covering outside the food. And why? Because it acts as a barrier to protect the food from the environment. Environmental hazards like water vapor, heat, pressure, so on, oxidation, ultraviolet rays, microbial contamination, even physical damage while packing or transporting. Inoculate the food, or you inoculate the food with the bacteriocin producer strains, or you have packaging films or you immobilize these peptides as a coating in the form of functional foods or nutraceuticals. And they also add the nutritional value. They increase the nutritional value of the food and exert regulatory effects on the human body. So the food packaging, besides preventing food spoilage, is also known to have increased nutritional value of the food that is packed or covered with these peptides, and they can also bring about a lot of regulatory effects in the human body. Microbial contamination, that is one of the greatest cause of concern, because that would cause a lot of food deterioration, and it can happen anytime. It may happen immediately after harvesting. Therefore, there is increased demand for antimicrobial food packaging, which is less with chemical preservatives and minimal processing. One of the reasons for antimicrobial AMP, as it is called, is because we are trying to avoid too many chemicals and minimum time of processing. So many biopreservatives of natural and antimicrobial origin have been used as polymers in the food packaging industry. One of them is, or rather, main one is the antimicrobial peptides. Antimicrobial peptides, they have a broad range of antimicrobial immunomodulatory activities against almost all the foodborne pathogens and infectious bacteria. How do they act? Is they interact with certain food components, and they prevent, such as they prevent the proteolytic degradation. But there are chances that these foods which we are packing, the AMPs which are used to pack the protein, the food, the protein or the fat inside that food itself may start acting with the AMPs, and they reduce their antimicrobial activity. So that is one of the dangerous side effects that can happen. And hence the AMPs are now encapsulated in nano carriers, so that you can have a sustained release, controlled release for food preservation during the storage, because we want to protect the AMPs against the self-degradation, which can happen by the own food. The protein and the fat can start interacting with the AMPs and break them down. So to protect them, we have them encapsulated. And there is kind of a controlled release active packaging, which is a very innovative technology. Next one. They were discovered in 1939 with from the Bacillus strain, and they were extracted as an antimicrobial agent from soil. These are basically the building blocks of the defense system in any living organism, found in almost all bacteria, plants, invertebrate, vertebrates. You can see ahead as we go along, even insects have these AMPs. The human cells of the gut also excrete AMPs, which protect us from pathogenic microorganisms. Next one. They have unique structural characteristics. They are amphipathic, that is dual nature. They can form loop structures. You know, proteins have the primary structure, the secondary structure, tertiary structure, and so on. So these peptides also, though they are small, they form loop structures and cyclic peptides because of certain disulfide bonds. There are a lot of cysteine residues, so you can have a lot of disulfide bonds. They show a lot of activity against gram-positive, gram-negative bacteria and fungi. In vitro studies were conducted. They are classified based on the pathogen that they are targeted towards, like antibacterial, antifungal, antiviral, or antiparasitic peptides. Sometimes AMPs are effective for 2 or more pathogens. It is not just one. They may be dual effective. Next one. How do they act? They have positive charge, so they get attracted towards the negatively charged membranes of the microorganisms. Most of them are hydrophobic, and they penetrate the core of the bacterial membranes, leading to a cytoplasmic leakage, disrupting the membrane, of course, and eventually causing the cell death of the pathogens. There is a World Health Organization, the WHO has identified certain antibiotic resistant bacteria. And you know, antibiotic resistant bacteria are always a major threat to human health. So many of these AMPs have been found to be very effective against such kind of harmful bacteria. Now, what are the sources of these peptides? You have various sources, as you can see, right from plants to cereals to insects, as I told you. So let's go ahead looking at the plants and the non-plant sources. From the plant sources, you have various groups of algae such as spirulina, chlorella, which show a lot of anti-cancer and immunomodulating activities. Marine microalgae also give rise to bioactive peptides, which have a lot of health benefits like cholesterol lowering, antihypertensive, immunomodulating, so on. Medicinal plants such as cannabis, hemp, that is hemp seed protein, acacia show antimicrobial peptides. So you have the different sources. Now there is this one particular enzyme, which is a major enzyme in photosynthesis and photorespiration in green plants. It's called as ribulose 1,5-biphosphate carboxylase or oxygenase, which is, you know, from spinach or alfalfa. Enzymatic hydrolysis resulted in smaller peptides with lot of bioactivities. So this enzymatic hydrolysis of this particular enzyme, which may have lot of amino acids as a long chain, when you break it down to smaller, after hydrolysis, you break them down to smaller peptides. They have shown a lot of multiple bioactivities such as anti-hypertensive, antioxidative, opioid-like activities, and so on. Now with the COVID, which is so recent, like now 2020, which we were all affected by COVID worldwide, globally, there has been a continuous efforts to get cure for COVID-19. So in silico approach, that is computational methods were used to identify anti-COVID peptides from microalgae like the spirulina. And by computational methods, which is called as molecular docking, they have been able to study some of the inhibitory effects of the peptides, these short chain peptides from spirulina, on the spike protein of COVID-19. It's a very recent work, 2021, and there is great scope ahead for this kind of study. But you can see that the plant source spirulina is leading towards giving you a smaller biopeptides, which may be a cure for many other, because you keep on hearing of newer and newer. One was COVID, now you have monkeypox, you have something else that is coming up, and there is no cure for it. But the food science technologists, along with the pharmacists and so on and so forth, they are all working towards different sources to find out how to bring about cure. So one of them for COVID was from the plant spirulina. Next one. The non-plant sources we are comfortable with, but also non-plant sources like, whey proteins. They have been hydrolyzed, and the peptides which are released showed a lot of health benefits for the immune and cardiovascular, nervous, and the gastrointestinal systems. Cereal grains such as wheat, barley, rice, rye, oat, millet, sorghum, they are rich source of bioactive peptides. Then you have certain curcubita seeds, which are valuable biopeptides in terms of their amino acid composition, very effective for, again, health promoting activities. Next. Egg proteins. Egg proteins are the bioactive peptides derived from egg proteins. They also possess antimicrobial, antioxidant, antihypertensive, anti-diabetic, and anti-inflammatory activity. Now, why? Because of the rich source of amino acids and the long shelf life, egg white protein has been used in the food industry and pharmaceutical industry as a shelf life prolongation. And most of the egg white protein is lysozyme enzyme, which is a rich source of biologically active peptides, such as giving you all these different, again, antimicrobial, anti-cancer, immune regulator, antihypertensive. So this has been used as hydrolyzed lysozyme, which is almost 6% in the egg white, is used in the industry as a natural preservative to prevent the growth of bacteria in meat products such as sausages, beef, and pork. And also the heat denatured lysoenzyme. Not only hydrolyzed, but the heat denatured also is shown to have good antimicrobial activity. Dairy products such as milk, cheese, from there also you can extract the bioactive peptides. Milk peptides have a lot of antioxidant, antibacterial, and ACE inhibitory, that is angiotensin converting enzyme, which is angiotensin is known to increase the blood pressure, whereas if you inhibit the enzyme with these kind of peptides, you can modulate the blood pressure and regulate it. Opioid peptides in milk, they have morphine-like properties on the central nervous system, so they can act like analgesics and painkillers. Lactoferrin also, which is present in milk of all mammals, plays an antimicrobial and immunosuppressive role. Antioxidant in whey hydrolysates is again due to the amino acid cysteine, which I told you has a lot of SH groups, and this helps in the synthesis of glutathione, which is a very powerful antioxidant. So you see products like milk and whey and these are very good sources for giving you a lot of bioactive peptides. Aquatic sources like shellfish, the industry is always looking out for newer and newer sources, because plant sources may be limited, but you're going into other sources like meat and fish, and now aquatic sources like shellfish. They are being explored now, whether they can be added as in functional foods, as nutritional supplements. Many anti-cancer bioactive peptides have been extracted from marine organisms such as sponges, mollusks, tuna, sardines. Venom peptides from snake venom also has shown to have antioxidant, antimicrobial, and hypotensive properties. So these are all the biopeptides coming out from the bigger proteins of these sources. Next one. Red meat, blood, they also have shown to have a lot of antimicrobial and antihypertensive activities. Other sources, animal sources, include rabbit leukocytes, lysozyme of human leukocytes, lactoferrin from cows, and low molecular weight antimicrobial proteins, peptides from the human female reproductive tract. So no source is left undiscovered. There are more sources being regularly discovered because the food industry is so large and they do need a large quantity of biopeptides. Next one. Bacillus strains also are known to produce AMPs, which show activity against Salmonella species, E. coli, Shigella, Staphylococcus aureus, and other strains of Klebsiella, etc. So even the AMPs that are extracted from Pseudomonas itself also shows some inhibitory. So it's like a dual, synergistic action where Pseudomonas AMPs will show an inhibition against Salmonella, E. coli, and Shigella, Staphylococcus. These are all known to cause food poisoning in different ways. Salmonella, typhoid, E. coli, dysentery, and stomach, GI problems. Shigella is known to cause dysentery. Staphylococcus is a known bacteria, gram-positive, that is known to cause food poisoning, simple vomiting to gastric problems. So you need to have some antimicrobial peptides, and which are sometimes they are from their own sources that you can get them from the different bacilli. Glycosin is a recent AMP that was discovered very recently, and it is stable at almost very high temperatures also. From Streptomyces, they have discovered salinamides, which are two bioactive compounds that have shown local anti-inflammatory and antimicrobial activity against gram-negative bacteria, as well as in the body to control some infections and to decrease tissue inflammation. How do we extract? There are various methods of extraction of biopeptides. This is a diagrammatic representation where you can see you have the conventional approaches like chemical, biological, or physical, where biological would involve essentially enzymatic hydrolysis or fermentation. There are novel or newer approaches, which are ultrasound, ohmic heating, microwave, pulsed electric fields, and subcritical water hydrolysis. So these are newer methods which are being used. Just to go briefly into some of them, which are being used today in the industry for extracting of bioactive peptides, because just to again recap what we said, peptides are mostly present in the original native protein. You have to get them out of the protein. They are within the protein structure. So for breaking down the protein and getting out these peptides, you need different methods. And the different methods employed to get these proteins out also should not damage the peptides. So therefore, they're encapsulated most of the times. And these peptides, they can go to the target tissue in concern and bring about the activity without leaving any trace or residual damage to the body, to the environment, or compared to synthetic compounds, they bring about very less of destructive activities. Okay, let's go ahead to the methods. Chemical method generally use sodium hydroxide. It's used to produce pharmaceutical peptides, like which are generally used for all infectious diseases. This process is very effective in obtaining most of the proteins or peptides in a soluble form, but it may cause a little bit of structural change, and that could lead to losing the original function of the peptide or the protein. For this, around 1992, there were radio waves which were then added to accelerate this method. So in addition to NaOH, the chemical method also used radio waves. Next one. Physical methods or the microwave extraction or ohmic heating, more economical, easy to use in the industry, like colloidal milling, homogenization, high-speed blending, freezing and thawing, high pressure have been used. Microwave heating is based on, as you know, electromagnetic waves, and it is very uniform, safe, simple, quick, and it's a very clean operation, besides low maintenance. It has lower impact on the food products, the flavor, which is very important. We cannot miss out that any method should not affect the flavor and the nutritional quality of the food, because then that purpose is lost. So even the AMPs or whatever you're trying to extract should not have any impact on all these aspects of the food. Ohmic heating was initially used for milk pasteurization in 1920. It's a thermal processing that alternates it uses alternating electric current directly into a semiconductor media. And it is very good because it reduces the heating time and it enhances the protein's emulsifying capacity. Next one. Enzymatic methods supposed to be one of the best. They degrade the cell wall, they improve the protein solubility. And these are some of the enzymes like phytase, xylanase. They have been used to isolate protein from rice bran. Some of the other proteases also are in use. In industrial scales conditions, the use of surface coated enzymes is more common than conventional soluble enzymes. You have surface coating. And why? Because the coated enzymes allow enzymatic hydrolysis under more moderate and controlled conditions. So if you have enzyme coated on the surface of the food, it would have it would be more effective, and it would be under controlled conditions rather than, incorporating them inside. Next. This is another method. Recombinant production is the expression of bioactive peptides in vitro expression systems called as a cell-free system, and very high speed. You can have mass production. So most of the pharmaceutical peptides are produced by this method along with the chemical. So you have chemical NaOH hydrolysis along with this method, so that you get the best mass production at a very high speed. One of the peptides which has been produced globally is a anti-diabetic peptide for type 2 diabetic patients. It regulates blood sugar, and this was approved in 2010 by FDA by this method, both combined chemical and recombinant production. Bioactive peptides, as I said, can be used in can be produced by bacteria in fermented food. Basically, lactobacillus species is the one that is used to produce these bioactive peptides. This method is very cheap. It is safe. It is of short growth time. The bacteria grow very fast, and these bacteria of lactobacillus, they're expressed on the cell wall so that they can be easily removed. It's inexpensive. So, these biopeptides can be then incorporated or surface coated on the food products. This is a newer method, pulsed electric field, and it has been employed as a non-thermal process for microorganisms and enzyme inactivation, where the food sample is subjected to the short high power electric pulses between two electrodes. The electrical energy that is generated causes the protein to unfold. You know that the protein is folded chain upon chain. So because of this energy generation, it causes the protein unfolding, enhanced interactions. It has been observed in treatment of canola seeds that it enhances the extracted protein solubility, emulsifying and foaming capabilities because of this kind of method which is used. And how? Because it changes the secondary structure of proteins, even in case of some other examples like pine nut proteins. So basically what it does is that it's a non-thermal process. And the food sample, whichever is to be protected, is subjected to this high speed, high power electric pulses. The food is kept between two electrodes, and the energy that is generated causes the unfolding of the protein. It has been tried out in many food examples. One of them is like canola seeds, where it was shown to have more solubility. The peptides have more solubility, emulsifying and foaming capabilities. It has also been seen in pine nut protein, where this change, because of the unfolding of the protein chain, cause more of antioxidant effect. Next one. Now, when you get these bioactive compounds, you naturally have to purify them. You cannot just use them. They have to undergo purification to become more commercial. So what are the methods for purification? You have reverse phase HPLC, size exclusion chromatography, ultrafiltration, ion exchange chromatography. All these can be used for purification. After purifying, you have to identify, for which you can have mass spectrometry, electrospray ionization, all the newer techniques which are used for identifying which are the peptides, what is the sequence of amino acids. Older methods used chemicals like Sanger's method and all. I'm sure you have studied all of them. But going ahead, technology has advanced so much that you can have more and more simpler methods, faster methods, which can give you the whole sequence of amino acids very quickly. Next. So these are different effect of different operational conditions on the functional property of bioactive peptides. These are all the different protein sources: dairy, eggs, cereal, meats. They are subjected to all these different degree of hydrolysis. Very important to keep in mind. So in the industry has to, have repeated trials to see which is the best method, which is the best pH, temperature, time, so that you don't get something which is not desired. You want something which is very good of good quality, ready to use, cheaply available, non-toxic, can be applied directly, can be sustained for a long time. So for that, they have to undergo all these tests to see what is the to see what is the degree of hydrolysis, how much of enzymes to be used according to the substance that is there, the type of enzyme. And then after it undergoes the proteolysis, you get the amino acid sequence, what is the molecular weight, and finally you get your functional bioactive peptides, which can be again subjected to sequential analysis. So what are the functions of biopeptides? You know that right from start, we have been saying that the biopeptides are not only related to health, but also in the industry, food industry. So how are they, where are they related and where are they used? As you can see, this is the center of bioactive peptides. They have various functions in our own body, anti-cancer activities, and the sources we have been seeing over the past few slides that they can come from various sources, right from plant sources to non-plant sources, from aquatic sources to venom to other different kind of, just go back to the other slide, please, previous one, thank you. So you have anti-cancer, antimicrobial, opioid activity in the central nervous system, immunomodulatory, hypertensive, mineral binding. So all these are different activities from these bioactive peptides, which are coming from plant and non-plant sources. Just to go back and revise, non-plant sources also are giving rise to bioactive peptides. Next one. So these are, once again, it's just to, highlight. These are the sources, as I said, enzymatic, giving you bioactive peptides, help promoting hypocholesteremic, blood cholesterol lowering, cardiovascular system, it improves, that is ACE inhibitory, regulates blood pressure, antioxidants, scavenges free radicals, boosts the immune system, antimicrobial in the digestive tract. Now, in the food industry, which is very important for most of you, it helps in the foam stabilization, solubility, emulsifying ability, water holding capacity, and surface hydrophobicity. So you can see the dual purpose of bioactive peptides. Next one. One very important effect is on the dental health and nutritional status, where at intestinal pH, alkaline pH, the peptides with specific sequences can bind to calcium, minerals like calcium and phosphorus, and they help in forming soluble organophosphates, leading to increased mineral uptake that prevents osteoporosis and dental cavities. Flaxseed proteins, which have a lot of hydrophobic and positively charged amino acids, help also in generating this protein, calmodulin, that helps in binding calcium ions. Milk casein also is known to increase the binding of calcium and phosphorus ions and thereby increase the bioavailability. So besides all the other functions that we have seen of, lowering cholesterol, maintaining the blood pressure, maintaining the other activities, they also help in mineral deposition and prevention of dental caries and osteoporosis. So here again, you have flaxseed protein, milk casein, which are sources of these bioactive peptides that are helping to health promoting activities in terms of dental health and bone health. They have, now this is a little bit of repeat, but as you can see, numerous bioactive peptides from food have been shown to possess cytomodulatory properties, increasing the immunity of neonatal intestinal cells. They are also from waste of the mozzarella cheese. They have anti-proliferative action. Casein-derived protein utilized in confectionery and soft beverages. Now, food industry may be using casein-derived proteins in these confectionery and soft beverages with the purpose of stress relieving characteristic. Sometimes you come across advertisements which say that, this drink or this beverage will help you in relaxing. Now, why? Because it probably would have these casein-derived peptides. So it is not something false that is being advertised. It is because of certain peptides which are known to cause stress relieving. They have these activities, so they are added by the food industry in all these products. Then you have in mammals anti-hypertensive peptides, which help in regulating salt balance, electrolyte balance. Then as I mentioned before, that milk-based bioactive peptides can be used to reduce the metabolic syndrome by modulating blood pressure, food consumption, and free radical absorption. So all these are again just clans of different activities of the biopeptides in our human body. Next one. How do you apply? What is the application of bioactive peptides in food technology, food safety, and pharmaceuticals? Next one. So here you see that there are different factors such as processing conditions, protein source, amino acid sequence, composition, molecular weight, the pH, chemical treatments. It will affect. All these are going to affect your bioactive peptides. And therefore, you have to have experimental procedures to actually standardize a method which you're going to use to get you the best source and the most stable source of bioactive peptides. The solubility of them depends on the molecular size. When they are hydrolyzed, when the protein is hydrolyzed to smaller peptides, the solubility, as you know, will increase. And the polarity of the amino acids will, of course, influence the hydrophil to hydrophobic balance, which will again influence the solubility. The denatured and hydrolyzed proteins have a good foaming ability. So therefore, you denature the proteins. Lower molecular weight peptides show more water holding capacity than the higher ones. And because they have more hydrophilic, protein hydrolysates from fish showed better solubility with a better, wider ionic pH range. And therefore, incorporating this fish hydrolysates to food products contributed to water holding capacity, which in turn improves the textural attributes, gel forming ability, whipping and emulsifying properties. Now, this is all related to fish, all related to the food industry. And to get better attributes to your food product, you can have proteins or the peptides getting, you can either get them from plant or, as I told you, non-plant sources. But of course, then you will have to label your food products accordingly with the green dot or the red dot according to the source of bioactive peptides which have been used to preserve. Otherwise, there could be some other legal issues that can come up. Yeah. Peptides are used as sweeteners, color stabilizers, anti-caking factors, thickening, emulsifiers, flavor enhancers, emulsifiers in food preparation and acidity control. They are also used to improve the food quality by affecting the water and oil retention capacity, colloidal stability, viscosity, and foam generation in the food finished product. So all these are attributes of these peptides, and these are all their attributes which can be made use of by the food industry. They are being used by the food industry for enhancing the attributes of the food and for extending the shelf life of the food. Next. Whey protein, soy protein hydrolysates were inhibitory to lipid oxidation in pork stored. When you're storing meat products. Casein hydrolysates were found to be effective inhibitors of thiobarbituric acid in ground beef. Ground beef and tuna homogenates had reduced the TBRS formation when egg protein hydrolysates were used. Production of peroxide was reduced in cooked beef patties by addition of potato protein and potato protein hydrolysates. See, we don't want the food product to go rancid. And most of these meat products and all go rancid very fast because of oxidation. And therefore, you want to have some antioxidant which can help in lowering these oxidative processes. And these are different bioactive peptides from different sources, which we have seen like whey, soy, casein, egg protein, potato protein. Buckwheat peptides are known to increase the shelf life of fish fillet, tilapia fish fillet. Peptide fragment from slaughterhouse delayed the meat rancidity by preventing lipid oxidation. Camel milk protein is also known to be effective in inhibiting lipid peroxidation. Then marine fish skin peptides were used to preservative in pork patties. Seafood are being used as antioxidant, cryoprotective, that is damage against very low temperatures, cryoprotective, very low temperatures. So they prevent protein denaturation during freezing conditions. Next one. Nisin is an antimicrobial peptide derived from the Lactococcus lactis, and it has been used as a food preservative for years. Whereas peptide fragments from slaughterhouse byproducts, so nothing goes waste. You're adding value addition to anything that is going waste of the food industry. They are
ये मैं अभी थोड़ा मीटिंग में हूँ, मैं कॉल करूंगा या मैं कल देखता हूँ, आता हूँ, अभी तो मैं बिजी हूँ.
They are obtaining the peptide fragments from these slaughterhouse byproducts, and they were known to inhibit microbial growth on refrigerated meat for almost 2 weeks. Emulsified and non-emulsified defatted raw protein from the fish inhibited microbial spoilage when applied to some sausages. Antimicrobial peptides were also used to reduce the microbial spoilage of fish like flounder fillet when they were applied on the fish. Next. Soy protein concentrate has been used in beef burger formulation. They improved the quality of the patty as well as the water holding capacity. And it increased by replacing beef with, you can also replace the beef with texturized. Nowadays, you have texturized vegetable proteins, which are simulating the meat product. It's not meat, or it's not the meat that is there, but it's a non-meat, it's a vegetable protein. So they can antioxidant peptides. The main purpose, you replace the artificial antioxidants because they have equivalent or even higher ability to suppress the lipid oxidation. So we are trying to aim at decreasing the use of chemical preservatives and rather going in for natural sources from plant or non-plant. And by applying them, you're trying to reduce the use of chemical preservatives. Next one. Foam formation can generate acceptable and textural sensory characteristics of food, such as pastries and sauces, and reduce the surface tension. So that is one of the attributes of these peptides. They can generate foam. Rice bran protein isolates had a very good foaming potential to egg white, but much lower foaming stability. So between the two, you can have egg white better, giving better foaming stability. Also, quinoa seed protein concentrate was much higher than soybean cake protein. So these are different sources which are being explored to give you better foaming characteristics, to get better gelling, to get better flavors, to get better texture. And they have found many sources, rice bran to egg to soya protein, etc. Soy protein concentrate minimizes the cooking, frying loss and shrinkage and enhances foaming stability in chicken sausages. So all different types of sources from where we have extracted the peptides, applied them onto these different sources to protect them and decrease the antioxidant, or rather prevent oxidation, lessen the off flavors that can be created, rancidity, etc., by using all these peptides from different sources. Also from certain Sardinella species, they have a lot of peptides with high antioxidant, especially for meat-based products. I've already mentioned earlier of dairy sources, so I won't go into this again, but they can be used for beef, and most of the lipid peroxidation is prevented, off odors are prevented, extending the shelf life. Next one. Pharmaceutical applications of peptides as anti-diabetic hydrolysates. You can add them to sausages. Now, food is always a medicine, right? So even from food, you can't say that because it is processed that it is a bad food. Processing is necessary. Processing is done even at the home level, because you see so much of, mention about processed food is bad, processed food by the industry is bad. But we forget that we are processing food even at a home level. And are we doing it exactly in the right way, and the right method is being used at the home? Can you ensure that the process that you use at home is much better than what is used in the industry? You cannot, because each home, each person will have a different way. And there is a lot of food wastage, a lot of loss of nutrients in the process of cutting, chopping, washing, cooking, etc. So when you add these biopeptides to certain food products and you're enhancing their medicinal properties or pharmaceutical properties, you can see that they can be used like anti-diabetic hydrolysates, which are added to sausages to fortify the sausages and to delay, or rather help in, lessening or keeping the blood sugar in control, because people they want to eat. And you cannot just stop them from eating certain things, but rather you offer them certain products which will help to control whatever problems they are facing. As I told you before, large number of peptides have been identified as ACE inhibitors. Even then, in infant milk formulae, ACE inhibitors is basically to control the blood pressure. Antioxidant potential of meat hydrolysate, chicken breast hydrolysate, rice protein, and tilapia collagen was also observed. Yeah, next one. Cosmeceutical applications, because cosmetics, they have been used in that industry also because of their bioavailability, stability, they are light, air stable, they don't have much toxicity, they show a powerful affinity for water and possess moisturizing abilities. They are very frequently employed in functional cosmetics to treat skin conditions, promoting collagen synthesis to keep one's skin looking young and, not getting wrinkles. Antioxidant, anti-inflammatory, anti-wrinkle, whitening, and wound healing. Neurotransmitter inhibitors, enzyme inhibitors, peptides can also be found as topical cosmeceuticals. They can if they are slightly larger, they can even penetrate the skin barrier, particularly for aged skin, dry and aged skin. So it helps in maintaining the firmness and the elasticity of the skin. Next. Facial creams, which are now gaining so much of importance. One of them, GHK copper, has got tripeptide. There were studies which were conducted for about 12 weeks on 71 volunteers in the age group of 50 to 59, and it showed a visible reduction in the sign of aging. Wow, why not? Okay, collagen peptides have been found to help improve the skin problems. They may prevent loss of moisture from the skin caused by ultraviolet light, to which we are all the time exposed, and therefore we use all the skin SPF creams when you especially go out. Wound healing applications. They are also used to stimulate the proliferation of keratinocytes, fibroblasts, monocytes, which are very important for wound healing. Collagen peptides have also got chemotactic qualities, which encourage cell migration, again for wound healing. Then there is a peptide which was discovered from wheat bread, which was enhanced with the addition of Palmaria palmata seaweed, which had renin inhibitory properties. Now, this also related to blood pressure and so on. So there is a new peptide from sesame seeds, which is used in a beverage called as Foshu, and this is known to have anti-hypertensive properties. Next one. Feed applications in the fish meal, or in poultry feeds. Animal proteins have been used because they increase the nutritional quality and they decrease any anti-nutritional effects. Aquaculture, all these are using these biopeptides in different feed applications. Next. Safety of bioactive peptides, very important. They have to be very safe. So for that, you have to have clinical studies, food applications, physiological impact of bioactive peptide consumption is thought to be harmless, but most of them need to have toxicological investigations in vitro and in vivo. Just a few investigations on the potential of toxicological impact on human has been undertaken. So it is very critical to have more studies on human safety where you can test for allergic reactions, toxic reactions, cytotoxicity. Strict and precise legislation is essential to safeguard the consumers from potentially hazardous or deceptive products. The market is flooded with good and bad products. So here you have to be very, very careful. Preclinical and clinical studies are needed to determine which levels are beneficial for health, dose response relation, bioavailability, pharmacokinetics, and whether they can be consumed with foods. Next one. This we'll not go into because most of it, safety concerns, same. Intake length, intake dosage, regulatory approval, intake frequency, stability. So these are all the different peptides, how we get them out, consumption of biopeptides, but we need to be very careful about all these concerns. Next one. What are the challenges ahead? Next one. Traditional extraction methods using chemicals have been widely applied that could affect environment and human health. That is one challenge. Now, challenges are going to come up anywhere and anytime. Even if as technology advances, you have challenges. You have challenges using your phone. You have challenges using your, all these technological methods. Everybody is facing challenges, but you have to overcome them. Activity of bioactive peptides can be affected by various factors such as processing conditions that alter their properties altogether, or even their bioavailability may be affected. So that is one more challenge. So the challenge is to retain the biological activity, like all these antioxidant, antimicrobial, functional properties in the food industry, especially during extraction, processing stages. Therefore, you can maintain their health promoting benefit, as well as they should be they should be safe enough to be applied to the foods, and they should remain unaltered and easy accessible. Some peptides are susceptible to degradation during intestinal digestion. In vitro studies have revealed that an anti-hypertensive peptide that was obtained from pea protein, it withstood pepsin digestion in the GI tract, but it was affected by the pancreatic proteases. So therefore, it leads to reduced bioactivity in some of the peptide fragments. So these are some of the challenges which are coming up, and these can come up only with a lot of research. So for young students, there is a lot of research scope for research in this area, where you can work with different biopeptides on different models, in animal models, in vitro, in vivo studies, and find out the best way to overcome all these different challenges. Next. Recently, a lot of different alternatives have been suggested, like using green extraction technologies, biotechnological approaches, non-thermal techniques such as high hydrostatic pressure, supercritical ultrasound, microwave assisted extraction methods. But they are very costly, so sometimes may not be economically feasible. Most common is the enzymatic hydrolysis, and it gives desired activity, plus it gives multiple, it requires multiple fractionation processes, but it gives the peptides with desired bioactivity. And sometimes the in vitro may not be effective in vivo. So it's very important to study both the models. Next. Now, one more challenge, very, very important, some of these, that is, there's a time lapse between food processing, distribution, consumption, at which point the bioactive peptides may interact with the food components, leading to deterioration of their biological potential. So that is one very important thing to be looked at. They may have some potential toxicity and allergenicity, which can be released during the hydrolysis. Another point, toxic effect. Bioactive peptides can be, sorry, the earlier one. Therefore, they can be treated with stabilizing characters. Encapsulation is one method where you can overcome these problems related to the direct application of peptides in foods. Why? Because it can keep the structural integrity and functionality of the peptides in the food matrix. There are concerns about the safety of peptides in food application, because as I told you before, there could be toxic from their parental protein. Something which is very different may come up. The parent protein may not be as toxic as the peptide protein. And hazardous components can be produced during this extraction processes. So these are some of the challenges, and way to overcome them is by encapsulation. Next one. Commercial production and application of bioactive peptides are limited due to several factors, such as suitable large-scale production technology, high production, cost, taste, and sensory quality of the end product. Next one. What is the future ahead? To improve the taste of peptide-added foods, you use masking agents like sucralose, fructose, and all these different masking agents, sodium acetate, MSG, sodium gluconate can be used. Micro encapsulation of peptides using a suitable carrier can be adapted to cover the bitterness. Spray drying using carriers such as soy protein isolates, maltodextrin, gum arabic, gelatin. It causes the bitterness producing hydrophobic groups to entrap, so the bitterness is reduced. High hydrostatic pressure processing is being used by the industries as a combination with protease treatment for acceleration of the peptide generation. And for continuous protein hydrolysis and isolation of multifunctional peptides, you have enzymatic ultrafiltration membrane reactor coupled with chromatography technique is being used. That gives you a very good, efficient, and very fast reactors to give you more and more biopeptides, and which we have looked at pulsed electric field hydrolysis is being considered a promising alternative for peptide production because of its low energy requirement, non-thermal, high efficiency compared to many other traditional methods. Next one. Advanced computational methods such as BLAST. Now, BLAST is also used. It is a very long terminology. It is called as Basic Local Alignment Search Tool, where it compares the biological sequence information and to the database of similar sequences. You know, you're comparing it. And you have in silico approach of bioinformatics. These are all advanced technologies which are being used to study the biopeptides. Uh, Underlying principle behind the in silico process, which is being used for that COVID, biopeptide, is to use available data or the information derived from molecular structure and implementation of the statistical and computational techniques for production of effective biopeptides. So you have a theoretical framework, and then that enables you to predict the potential bioactive property of the peptides, identify the peptide of interest within the protein, choose the enzyme with the desired action specificity, and then elucidate the efficacy of a protease either alone or in combination for releasing the bioactive peptide from the protein, from the parent protein. So you have methods of use, finding out which is the best enzyme to break down. From the main protein, you get the peptide of importance. And then, of course, you have to purify it, study its sequence, and all those other procedures have to follow. Use of inexpensive protein sources such as meat, marine products will be not only cost effective, but it will reduce the cost of waste disposal or incineration. So you have to look at that also. So therefore, insect proteins have been recently used because they are sustainable, viable, high quality proteins comparable to milk proteins. So microalgae, all these are different types of sources which are being looked at now. Employment of immobilized enzymes would allow reuse under different working conditions. Continuous hydrolysis or fermentation methods will give you better efficiency, lower operating cost compared with conventional batch process. That means you don't have to do batch wise, but it's an ongoing continuous process hydrolysis, which will give you more efficient and more number, but batch wise it will increase. For large scale continuous production of bioactive peptides, enzymatic membrane reactors coupled with chromatographic technique was recommended. And for the selective isolation of bioactive peptides, you can have combination of separation mechanisms of electrophoresis, membrane filtration, which is called as electromembrane filtration. Selection of a suitable food product as a carrier of bioactive peptide is very important to ensure that the consumption of food by the target consumer is tolerated and it will protect the bioactive property during the processes. Baked products which are widely consumed and which are don't have so much of processing conditions can be excellent sources for bioactive peptides. Or you have yogurts, yogurt like fermented products where number of bioactives can be added. Hypertensive peptides also were added. They were not affected during the shelf life. So this is all industry applications where they can look at different sources to extract the biopeptides by using simpler, I won't say simpler, more newer methods, more newer technologies, but which would give you mass production in shorter time and give you the purified, analyzed, sequential biopeptides, which could be then applied and incorporated into different foods for extending the shelf life. Next one. I won't say conclude because I just want to add a little more here. It's not there in one of the slides. Where all are the functional, where all are these biopeptides being used? They are being used from as functional foods and beverages. The protein hydrolysates basically from milk, soy, fish. They are incorporated into functional foods and beverages, as you know, for health benefits. Casein-derived peptides are very popular in the dairy industry and in beverages for heart health. In sports nutrition, now those of you who are into sports nutrition would also be very happy to realize that the bioactive peptides are being used in sports drinks, protein bars for their role in muscle recovery, immune boosting, and for nutrient absorption. They are used in meat and fish peptides to preserve the shelf life extension because of their antimicrobial and antioxidant and texture enhancement properties. Dairy products like yogurt, fermented milk, they are enriched with bioactive peptides to enhance the health claims. Health claims, as you know, are all different. Anti-hypertensive, anti-cancerous, so many, all anti, but all health benefits. Lactoferrin and casein peptides, they are known to boost the immunity and anti-inflammatory. Bakery peptides, which we just saw added to bread, biscuits, also improve the nutritional profile of the foods. Wheat and soy derived bioactive peptides are very strong antioxidant. And probiotics, now so much of probiotics, prebiotics, prebiotics. So all this prebiotics, probiotics, and fermented foods, especially like yogurt, kefir, kefir is like a fermented milk. Sauerkraut, that is a finely sliced cabbage, which is very popular in the Eastern European communities, in Southeast Asian communities, where they allow it to ferment with certain lactobacilli, microbial enzymes, which release the bioactive peptides. And these, on consumption, they are known to have a lot of health benefits, gut health, especially gut health and immune boosting functions. So besides all that we have looked at so far in the extraction, sources, purification, use in the food industry, you also have newer uses now in fields like sports nutrition and pre and probiotics in treatment for something like COVID and all the different diseases which are now newer and newer diseases which are coming. It's a promising future for the bioactive peptides. Because of their highly sensitive nature of the amino acids, you have to be very careful. Of course, you have to see that they are not toxic and they do not give rise to certain compounds like nitrosamines, acrylamide, when they interact with the food matrix. So all that has to be taken care of. Secondary metabolites also may affect these bioactive peptides, and therefore they are considered to be beneficial only in appropriate doses. High doses, when you say something is good, people tend to have a lot of it. There is no stopping them. You say flax seeds are good, so everybody jate hai and flax seeds kahan se bhi kharidte hain, and they use it in any way without knowing what could be the anti-nutritional properties in them. So for a lay person, the moment you say something is good and it's easily available across the counter, there is no stopping to the quantity that can be consumed, not knowing that the good thing can also become harmful to the human body. So same thing with the bioactive peptides also. They are appropriate only, they are beneficial only in appropriate doses, and their consumption at higher doses for their long period is quite dangerous. There could be populations which are sensitive and may cause severe adverse effects. Like, say, for example, vitamin A. Vitamin A is very good, but if you go on consuming a lot of vitamin A capsule that were fish liver, cod liver oil, they are fat-soluble vitamins and they can remain in your liver and other parts of your body, and they can be very toxic. Hypervitaminosis. So same thing. Vitamin bola to, vitamins are no problem at all. So don't go by that logic. Not you, I'm talking of the lay persons, but it is our role and duty to spread the knowledge of these very important active compounds, which you can get from proteins, but how much to be consumed and how to be consumed is our job, your job as young people here. And I'm sure many of you are looking forward to getting into the food industry. And AFST as a body is there to encourage you, help you, mentor you, and guide you on the right path to, further your career prospects, to enhance your career prospects. They are all there to help you. And I'm very thankful to AFST Mumbai chapter for giving me this opportunity to be here on this Sherpa, the educational part of it, to be there and share whatever little knowledge that I have with you on this very important topic. So thank you all. Thank you very much. I hope I've kept to the time. I don't know.
Hello. It was wonderful lecture, Dr. Geeta. I'm happy I could be late, but I could join. Though I was late, I could join and enjoyed your lecture. Thank you very much. All the best.
Thank you.
I'm
Dr.
little
Kondekar
outside.
is my mentor. So we learn from you. So thank you so much.
Thank you.
Always look forward to listening from you.
I'm actually outside.
Thank you so much.
Yeah. Asta, can you please take the questions? Ma'am, there are few questions in the chat box, so Asta will take one by one. So if you can answer the questions of the students. Asta will ask you the questions. Asta, are you there?
I can read the questions. There are two questions here.
Yeah.
Challenges for extraction and purification of bioactives, but other than these challenges, are there any other challenges for widespread acceptance? Well, anything new is always a challenge. Like when we talk of genetically modified foods, that's still a challenge. That's still not acceptable by many. So likewise, anything that's new. I wouldn't say bioactive peptides are new, but yes, there are newer methods that are coming, newer sources which are being looked at. And with that, there are newer challenges definitely to be faced by the food industry. How are you going to make them acceptable to the common man? How are you going to make them acceptable to the to be incorporated with the least damage to any body functions or any damage to any other environmental to your environment? So definitely these are challenges for the acceptance and the usage, and of course the cost, because when we are looking at newer methods, newer technologies, that is going to come along with a higher cost, and for higher cost the product also will be likewise priced higher. So there is definitely going to be a cost factor which is going to be a inhibitory to the wide usage, but that does not deter you from going ahead in this industry and developing more and more bioactive peptides. Definitely not. That challenge can be overcome. And then there is any commercial application of using plant bioactive in food preservation. Yeah, I didn't mention all the commercial, right from cosmeceutical to pharmaceutical to food industry to bakery industry to meat preservation to extending the shelf life. Okay, so all that is very definitely there. These are all the commercial applications of bioactive peptides. Yeah. What else is this? Of course, the PDF will be shared. That is with ma'am, Jyoti ma'am. Please share the PDF. That's not my private property. It can be definitely shared.
Sure, ma'am. There is one more question from Dilip. He's saying, thank you, ma'am, for the session. Can you please elaborate on the use of antimicrobial peptides in the industries for food preservation? Does the source of peptide influence is used in food preservation?
Antimicrobial use of antimicrobial AMP in the food industry. Yes, there are so many uses which I have listed earlier in right from preservation of extending the shelf life to the texture, the flavor, the sensory attributes of different food products, starting from the non-vegetarian, like beef and fish fillets, the beef, the patties, the burger patty, where they are more prone to rancidity of flavors. They are definitely, the application of bioactive peptides is going to increase the shelf life. It may increase, as I said, the cost also. And so that is one of the uses. Besides, as I told you, they have a lot of health benefits. So they are being used in probiotics, prebiotics, which are now known to have, in the process of fermentation, dairy products. All these are different sources from where you're getting these bioactive peptides, and they are known to either be used in the dairy itself, or they are being extracted from the dairy and being applied to other food products, to even plant sources. We have seen so many plants are also giving rise to bioactive peptides from where they can be extracted, like spirulina plant, which has given rise to this COVID cure for COVID they're trying to look at. So definitely applications are vast. Does the source of peptide influence is used in the food preservation? Well, yes, it would influence. Just give me 1 second if you don't mind. I'm getting one urgent call. Just 1 minute. I'll just. Sorry about that. This is what happens when you're at home sitting online. If I was in a classroom, I wouldn't have been attending to a call. Okay, anyway. So does the source of peptides influences use in food preservation? Yeah, it does influence. It will influence to a large extent, because whether it's from a plant source and it's a vegetarian source, easy to extract, whereas compared to a non-vegetarian source from where the source itself may be difficult to get, like, they're looking at fish, they're looking at venom, they're looking at insects. So imagine all these sources to get them and then to extract them and then put them as a preservative in food preservation, label that food as with a red dot. So it is going to have its influence. It may not affect the textural or the sensory qualities. It will not affect. That is taken care of. But other things definitely it will influence. And what else is there? Yeah, I think it more or less. I hope I've answered your queries on that. And I'm so happy to see so many of you here. I don't know who all. Many of you may be students. Some of you are my seniors, as I can see. Dr. Kondekar is there. He's my mentor, and I'm humbled.
Vinayati ma'am is there, also there, ma'am.
Vinayati ma'am is there. Oh my God, Veena
Yeah.
also got it. Veena is my all-time mentor, friend, and what should I say? Supporter. She gives me the confidence, so I'm so happy that she's here. So
Great,
yes,
great.
thank you, Veena, and thank you, everybody. I don't know, I've missed some of you who are there from the industry or any other field. And thank you, Jyoti from ICT. ICT has been like a second home to me. Over the years, we have been there. In fact, I started my PhD career there with Dr. Rege, but unfortunately, it was a time when I could not continue. He encouraged me to do my MPhil, complete it, and then come and do your PhD. So I did partial work with Dr. Rege at ICT. So I have memories, wonderful memories of ICT labs and all that. So from there, I've met so many of the ICT members who are my friends now. But then I moved on to something else and did my PhD in some other clinical work. So anyway, but ICT has always been a favorite. And now it has become such a big place to go to. So we look forward to interaction with all of you. And I don't know where.
We just on 16th of October, we had Professor D.V. Rege Memorial Lecture.
Yes, I know
so
that.
I must say that we missed you there.
Yeah,
so sometime.
I miss all the programs because I'm not in Bombay.
Okay.
I'm kind of out of Bombay. I'm based sometimes in Mumbai, sometimes in Kerala, where my husband is. And then at times I'm out of the country. So whenever possible. So yesterday, since I was there, I made it a point to attend PFNDI. I was so happy to go there. So,
okay, great.
if you'll update me on of course, you all put it on the post whenever you'll have these seminars and all, I'll try to be there definitely. I look forward to. Yeah, thank you so much. Thank you all.
Great.
And I hope I've kept
Veena ma'am has, she is there if she would like to say something. Veena ma'am?
Veena ma'am is there still? Oh yeah, she's there.
Oh yeah, hello. Yeah, very good afternoon to you all. Actually, I'm out, so I could join only for last 10 minutes. But Geeta is like our mentor for everything and biochemistry, and she has done extensive work in this field. So I am sure all the students are very happy because this particular kinds of compounds have a lot of application. So well done. Congratulations, dear Geeta, and also to Jyoti madam for organizing such wonderful sessions. I mean, it was amazing. Really very nice. Missed out. I would love to have the recording of the same. Thank you so
Ma'am,
much.
we will be uploading all the sessions on our YouTube channel of AFSTI Mumbai chapter and CASMB2. So, and
Dr. Jyoti.
For
Yes,
sure.
sure. Thank you so much.
Hello, Dr.
Thank
Jyoti.
you,
I have to say
Veena.
something.
Thank you.
Yes.
Hello, Dr. Jyoti.
Yes, sir.
Yeah. Other day I could not join the AFST Mumbai chapter program. But I had invited Nirupama, daughter of Dr. Rege, personally. Yeah, and she has given compliments and thanks to you for arranging this lecture.
Yeah, thank you so much.
Sir. Okay.
Great. So I think with this, we come to the end of the session. We thank you, Geeta ma'am, first of all, accepting our invitation, taking out the time for this lecture series. I'm sure the students have been enlightened to listen to you, all these sources, different methods, application, challenges of the bioactive peptide. And we can say that it is one of the important topic where the research should be there more and more. So thank you so much on behalf of the AFSTI Mumbai chapter, CASMB and ICT Mumbai once again. And I hope if the students have any questions, they can come to talk to us, write a message, and we will get back to you through the Geeta ma'am for sure. With this, we just want to have the another announcement. So, Geeta ma'am, the next Saturday we will be having the lecture by Mr. Sahil Desai on vitamin D. He is the Vice President International Business Development and Marketing at Fermenta Biotech. So, we appeal all the students, do not miss the next lecture. I am sure as you have enjoyed today's lecture, you are going to enjoy the next lecture too. So with this, we thank Geeta ma'am, all the audiences, Veena Yardi ma'am, Kundekar sir, everyone for joining the session.